Good afternoon, everyone. Thank you so much for joining us. We're going to go ahead and get started. I want to welcome everyone who's in person and online. I'm Keisha Pollock-Porter, the Dean of the Bloomberg School, and I'm so excited to welcome you to today's Dean's Lecture. This series is an opportunity to highlight the work of newly appointed and promoted professors and senior scientists. During these lectures, we have a chance to hear firsthand how they're changing the world through the power of public health. Today, I'm delighted to introduce Kasper Daniel Hansen, an international expert in statistical methodology and scientific discovery in molecular biology. Kasper is a professor in our Department of Biostatistics, but since joining the faculty 14 years ago, he has extended his influence and work across the school and university. You might want to take a while. This is a little bit long. I got a lot. You're doing a lot of great stuff. So, he has joint appointments in human genetics and biomedical engineering, and strong links to the Center for Computational Biology. He's a leading thinker in the areas of epigenetics, genome structure, and transcriptomics, and in particular, the statistical methods needed to appropriately analyze and interpret data relevant to those topics. This work is really fundamental to helping us understand how cells behave and is a powerful step toward finding new ways of preventing disease. Kasper earned his master's at the University of Copenhagen and then came to the US to pursue his doctorate at UC Berkeley. A quick, fun story from that time, and I want to thank his colleague, Margaret, who also happens to be his wife. So she gave us a little bit of background, which I thought was very interesting. So Margaret, if I say anything you don't like, please look that way, not here. So what we heard was that when Kasper was growing up in Denmark, apparently it was common to use English swear words in casual conversation. So as a PhD student who was new to the US, we heard that Kasper would regularly drop a few F-bombs in his presentations. I think this is great. Thank you so much. Eventually, Kasper was told this wasn't exactly the way we tend to communicate in scientific settings. And I'll just say, although Kasper may have had to adjust his language a bit, he has not lost his passion. Among his colleagues and students, Kasper is appreciated for his candor and his straightforward feedback. It's a great quality that he's not afraid to share what he's thinking, and he is known for approaching biostatistics with true rigor and passion. Kasper cares deeply about good science, and you can see the results in all that he has achieved. Kasper has had tremendous success in developing and applying statistical methods for genomic data and using data science approaches to make discoveries in biology. More specifically, he's had an interest in bias and noise in high throughput data and how these are related to specific steps in an experimental protocol. In other words, Kasper is helping us to make sense of massive amounts of genetic data and what they can tell us about diseases. He builds tools that help researchers find telling patterns while also identifying mistakes and random fluctuations. Ultimately, his work helps turn big, messy data sets into biological discoveries that we can trust. Through methodological contributions like all that Kasper has achieved, he is providing powerful, flexible tools to advance studies that improve human health. He has steadily emerged as an international leader with influential publications in journals such as Science, Nature, and Biostatistics. He's won so many honors, including the R35 Maximizing Investigators Research Award, which is a prestigious, highly selective NIH grant given to a person, not a project, in support of their exceptional research. As his colleagues will tell you, Kasper is also devoted to making the department and school a better place. He is a generous and supportive teammate and mentor who inspires others with his commitment and curiosity. He has been known to sit with the student or trainee for hours trying to get to the bottom of an intriguing result. Kasper, I want to thank you for your deep commitment to your work and to those around you, and we're so grateful that you contribute to the field and to the school. We're excited to hear from you today. I also want to welcome Kasper's family who's in the room and also online. Professor Hansen, please come to the podium. Thank you.
Yes. So thanks a lot for the nice words, Kisha, and thanks for everybody who is attending and spending some time on this. So when I was invited to give this talk, I was a little nervous. I've never done it before. So I asked a bunch of senior faculty in my department and sort of previous in the department who gave pretty useless advice. All of them, actually, it was pretty striking. All of them said, well, there's been a lot of unusual talks from this department in this lecture series, so there's no way you're going to stand out in a negative way. And then that was it. Except for one person who is going to remain nameless, who looked at me and said, I worked really hard on my talk until it was perfect. Okay, so I'm going to talk a little bit about the work I've done in statistical epigenomics, which covers a part of what I've done as a faculty, but not everything. So let's talk a little bit about what these words mean. So I'm really using statistics to help understand molecular biology. And sort of the statistical part of statistical epigenomics means that I analyze data, and I also develop sort of new methods to do so. Epigenomics is the study of all epigenetic changes in a cell. It doesn't really tell us that much because I haven't defined what the word epigenetics is. That's for the next couple of slides. But it kind of hints at sort of high-dimensional data because we are studying a lot of things at the same time. Okay, so one of the reasons why so this talk here, I've worked on a lot of different things, like Kisha was saying, been pretty interested in bias and problems and so on, but this is sort of more talk about discovery. So in epigenetics and in biology, one of the fundamental questions we have is what is the basis of phenotypic variation? And here we have members of two different species, and if you ask sort of a card-carrying biologist, not me, what is the basis for the phenotypic variation, they will say, this is genetics, even though these two species have very similar DNA. They are 99% of the genome is identical. That remaining 1% really encodes sort of the difference between the species. This is for the people who remember. This picture is also in my job talk, so we're going sort of full circle here. You can also ask, what is the basis for phenotypic variation sort of between organ systems or between tissues? And from a certain perspective, I will say that these two different organs we have here it's a little hand-wavy, but perhaps they're even more phenotypically different than the chimp and the human we saw on the previous slide. And if you ask, what drives the difference here, the answer is definitely not DNA, because these two organs, if they come from the same person, have exactly the same DNA in all of their cells. So there has to be something that encodes kind of organ specificity or cell specificity. So this is often used as sort of a definition of epigenetics, and some people would call it sort of heritable changes in phenotype not caused by DNA. Here, I mean heritable over cell division, so within an organism. I'm not talking about germline heritability, or the heritability most people think about when they talk about heritability, which is sort of heritability between people. Another way of saying this is that we study how cells with the same DNA acquire different identities and behavior as they develop. You can kind of say that this is sort of an information-focused definition of epigenetics. It's sort of focused on how information is transmitted between cells during development and differentiation. But there's other definitions. This is a word that means a lot of different things to different people, which basically makes the word kind of complicated to use. And that's true for the current literature. Different authors use it to mean different things. And for a really lengthy exposition, you should see this pretty nice book by John Greeley, a colleague, who wrote a bunch of opinion pieces about this in different scientific journals, and then basically was fed up and was like, I'm going to have to write a whole book about it because this is how complicated it is. It's pretty fascinating, actually. So it's kind of one of these bad things where we don't know exactly what we mean, which makes it ripe for overinterpretation. So when we say epigenetics, a lot of people will also talk about chromatin. So we have DNA. DNA is inside all our cells, and DNA is essentially a really long molecule. If you take DNA and you stretch it out from different cells, it's about 1 meter long, and you have this ginormous molecule that has to fit inside each individual cell. And the way you do that is that it's sort of packaged up a little bit like a ball of yarn. So it's packaged up into something called chromatin, which is a complex of DNA and proteins. And sort of major players in this process is something called DNA methylation, which you can see over there where it says methyl group. That's a little chemical addition to the DNA. And another major player is histone modifications, a histone. So you can see the DNA, the purple band, is wrapped around these blue balls that are called histones. And histones have these tails that you can see out, and some of the tails have this like epigenetic factor that's a histone modification attached to them. So a lot of people in the field sort of equate epigenetics with chromatin, but that's not actually true if you follow this information theoretical point of view. In this talk, I'm really talking about chromatin though. I'm going to talk a little bit about the 3D structure of the genome. I'm going to note out that on the left, up in the top there, we have this X here, which we can see through a powerful microscope. And at the bottom, we have this like very high resolution thing with like the DNA wrapped around individual proteins. And we have a pretty good idea how the major look at, how this sort of zoomed in look is. But all this stuff that happens in between, this like really big complex and these small things are actually not that well understood. You see it on a lot of graphs, including this one here. And typically, it's sort of artistic freedom. We don't really know what is going on there, at least as far as I understand. Okay, so information transfer is not just about chromatin though. If you've ever seen a video of cells dividing, it looks a little bit like a water droplet splitting into two water droplets. And that's a physical process, right? So we know that the mother cell has a bunch of molecules, proteins, and RNA, and also DNA, but certainly also protein and RNA and other stuff that are inside the cell. And the cell gets divided up into two daughter cells, and all of these molecules have to go somewhere. They either go to one of the daughter cells or the other of the daughter cells. So that's sort of direct information transfer that in some sense have nothing to do with chromatin or DNA and so on and so forth. So in reality, information transfer is sort of much harder to think about. And also, interestingly enough, even though it's part of the sort of the classic definition of epigenetics, at least according to my training, it's not really something that we operationalize very much when we do experiment. It's hard to measure information transfer, so we are often doing sort of other stuff that really have nothing to do with the definition. Because it's a misused word and can mean a lot of different things, there's been a lot of interest in epigenetics and gets attached to a lot of things. So for example, now you can buy epigenetic shampoo, which I'm not really sure what it means, although it does have 5 stars. So it gets reused or misused quite a lot. And I think this is sort of important to think about in these times here, where we're almost living in sort of a post-truth world. I'm going to talk a little bit about the end of the talk. It's also for sure being used by biohackers and people in the social sciences who love to sprinkle a little bit of biology around, even if it doesn't really make a whole lot of sense. Okay, so now I'm going to talk about sort of 3 different stories about epigenomics. First thing is sort of an old story about A and B compartments. Okay, so I talked about the 3D structure of the genome. And the main way we study 3D structure of the genome in biology is through an assay called Hi-C, which is a really expensive assay. Because it's super expensive, you usually have 1 or 2 samples in your paper, and you run into Cell or Science or Nature. But it's like very, very expensive on a single sample. What you get out of a Hi-C experiment is a matrix, and the matrix is sort of rectangular, and on the rows we have a chromosome, and on the columns we have a chromosome. And each entry in that matrix, it's a symmetric matrix, measures kind of how frequent 2 positions on the genome interact in 3D space. So it's kind of a projection, it's kind of a representation. When you look at the Hi-C matrix in sort of an unnormalized form like we have to the left, we really only see like a very high intensity around the diagonal. And that's because when things are really far apart along the linear genome, they tend to only occur sort of not that frequent. Most of the interactions are like right next to each other. So we see this sort of exponential decay of interactions over time, but that picture on the left is like not that interesting to look at. So in the first paper from 2009 that used this, they introduced something they called the observed expected normalization, where you calculate the expected interactions, the expected level of interaction for 2 loci that's separated by a given distance in linear space and normalized by that. And then you see this sort of plate pattern coming up. So in the first paper where they saw this, they basically concluded that this plate pattern, which they can describe through that vector below, you can see when it's red or when it's positive, it's in one part of the pattern, and when it's gray or negative, it's on the other part of the pattern, and technically it's the first eigenvector of this matrix here. It basically divides the genome into two compartments or two domains, one which is primarily open and active, and the other one which is closed and inactive. And typically, interactions occur mostly within domains. And because they were not very good with words or ideas, they called the two domains A and B. Okay, so this here is sort of one way of describing this. Of course, you can do a lot more with high-C data. People are really interested in like high-resolution interactions, and I'm sort of skipping over that, or I'm not talking about that here. So this came out in 2009, and we were working in my lab, especially with my first graduate student, Jean-Philippe Fortin. We worked a lot on DNA methylation data, and we started to work a little bit on high-C data. And we noticed that we could do this trick here. Well, I'm not really going to explain in detail what the trick is. But at the top, you have that eigenvector that I displayed in the previous picture, stretched out a little bit. That comes from high-C. And at the bottom, you have a function or a vector that is computed based on the correlation matrix of this DNA methylation experiment. So what we do is we take a completely different type of data called DNA methylation that is pretty easy to profile in a lot of different people. So we profile it in, let's say, 100, 200 people, and then we compute a sample-to-sample correlation matrix across the genome. Then we take the first eigenvector, and then we do a bunch of other tricks. It's not just like computing this. And then we get this vector up here that bears a striking similarity with the top vector. This is something we kind of realized just by playing around. This is sort of purely undirected discovery. This was totally unanticipated and pretty surprising. And it basically happened because we were playing with these two types of data at the same time. You can ask a little bit like, why is this happening? And of course, you can always ask why. We have a lengthy discussion about that in our paper. But at a certain level, on the left here, we have the high-C data matrix sort of normalized with this observed to expected ratio. On the right, we have the correlation matrix, sort of processed correlation matrix of the 450K data. And you can see how we have this plate pattern on the right. We have some loci where there's high correlation, even across long distances, and some loci where there's low correlations. And if you look really carefully, you can see that the high correlation boxes correspond to correlation between loci in the closed domain, and the low correlation thing corresponds to correlations in the open domain. And this is sort of how you get this data matrix down there. So this work here sort of established a new and quite surprising, I will say, link between DNA methylation and the 3D structure of the genome. There's been some follow-up work on this, like people have done 3D structure with various types of epigenetic data since then. But this paper here was really about sort of basic science, sort of curiosity-driven science. And sort of on a different note, I want to highlight that Stephen Cristiano, who was a graduate student in biostatistics, and then Rob Schaft and Victor Vulculescu later were pretty inspired by this. They were not really basing on it, but they were pretty inspired by this work here for their work on cell-free DNA fragmentation for cancer diagnostics that they afterwards spun out to a company called Delphi. Okay. So that's my first little story, and now I'm going to switch into something completely different. So perhaps I should ask if there's some questions here about the science, which there may not be. I'm not sure we have time to really dig into the details, but we can take questions afterwards. Okay, so the next little vignette is something I worked on for quite a while, which is sort of a broader thing called the Mendelian disorders of the epigenetic machinery. Okay, so in genetics, we are interested in studying phenotype. And we link DNA to phenotype through, for example, a genome-wide association study or sort of classic genetics. And that's, in some sense, really easy. And one of the reasons why it's really easy is because DNA doesn't change over time. It doesn't change from cell to cell. It's like constant in an individual. So we have a very clear causal pattern, causal diagram. We have a single arrow. It goes from left to right. Sometimes, or what we are interested in epigenomics and in many other areas of molecular biology, is sort of dissecting what goes on behind that sort of arrow line, what actually happens in the mechanism from DNA to phenotype. So we can draw a little black box, and we can fill it up with sort of epigenetic marks or expression or other things you might be interested in, protein, other things. The problem here is that these things change over time, and they change from cell type to cell type. Also, they're shaped by the phenotype. It's absolutely clear that we have some feedback loop from the phenotype back into expression and epigenetic marks. So in terms of causal diagrams, this is a complete nightmare. This is sort of one of the reasons why it's very hard to establish these things. It's one of the reasons why epigenetics is hard to work with, but the same thing is true for expression. There's a situation where the causal diagram is a little bit simpler. So Mendelian disorders are human disorders that are caused by mutations in a single gene. And Mendelian disorders are actually a really important source of knowledge about gene function. A lot of what we know about the function of human genes, we know from the study of these disorders. And a lot of this was basically started up at Hopkins by Victor McCusick over in the Department of Genetic Medicine, who's sort of the father, or well recognized as the father of of medical genetics. So what you could imagine doing is you could look at Mendelian disorders in genes that encode or kind of drive formation of chromatin. In that case, in those disorders, we have a very simple, somewhat simple causal diagram. We have a genetic defect, and if you think that the primary role of the gene is to shape chromatin, there's going to be some epigenetic defect, which depends on the cell type and the time at which we are measuring, and then that should cause a phenotype. So I got pretty inspired by this, by a paper written by Hans Bjørnsen. It was later followed up by some work by Hans and Jill, who sits down here. Hans is also here somewhere. And in that paper in 2015, he displayed this like a pizza picture. So let me explain what it is. It's a fairly complicated thing. It's like a pizza. There's a lot of slices, and there's a lot of small slices. And each small slice is a different gene. The pizza is divided up into 4 areas, and that's sort of 4 broad different groups of genes that have something to do with chromatin. They can write chromatin, they can erase it, they can read it, or they can remodel it or sort of move it around. What's interesting, what struck me when I read this paper by this wheel here is really the colors on the wheel. So the colors are phenotype. And I'm going to note that one thing that's sort of very striking is the yellow color out of the edge of the pizza that represents intellectual disability. And almost every single Mendelian disorder in this category has that phenotype. This here, Mendelian disorders of this type here is a class of disorders that are rapidly growing. Some of these have not really been described until recently. Actually, every year we find new disorders. So I think the latest is, how many do we have now? 100 or something like that? 100 approximately. The experts are sitting up here. But this, to me, tells us something really striking about the relationship between chromatin and phenotype. Especially, it really tells us that we can kind of hit chromatin with defects in sort of different directions. All of these genes do sort of different things to chromatin, and we still get sort of broadly the same phenotype. So I worked on this with Hans and now Hans and Jill for quite some years. And we have sort of taken different angles on it. One thing we started with, and we have to mention, got to take a little detour. So we can ask, what are chromatin regulators actually? And if you read the literature sort of historically, often it consists of some researcher, usually pretty well read and smart, sits down and says, these are the genes that I like. I'm going to put them on my list. So this is sort of the chromatin regulators. But in 2019, we developed sort of a more, I would say, objective way of defining the epigenetic machinery using protein domains. So we have systematically identified protein domains that sort of touch chromatin and say any gene that has such a protein domain is going to be part of the epigenetic machinery. So we have some interest in these Mendelian disorders, but I'm going to highlight that there's sort of a growing interest in these disorders in molecular biology, particularly in cancer biology. One of the main results from the Cancer Genome Atlas, which was a big effort on sequencing many human cancers, we now know that somatic mutations are frequent in these EM genes. It's a little complicated. It depends a little bit on the cancer type, which gene it is that's frequently mutated. But this really suggests to cancer biologists that there's something important about these EM genes, or these specific EM genes in these specific cancers. Another area where these genes have popped up is sort of neurological disorders. Over the last couple of decades, sort of GWAS and rare variant analysis has implicated these epigenetic genes in various neurological disorders, including schizophrenia and autism. In fact, most of the most well-recognized genetic causes of autism are on this list of epigenetic disorders. Okay, so Hansen, me, and Jill has worked on this from sort of different angles. And now, since we are in public health, I got to talk a little bit about potential treatment, although it's going to be nice. So one of the first things I was involved in, and I was really more of a hang-around at this time here, was an attempt at treating one of these disorders called Kabuki syndrome, which is caused by a loss-of-function mutation in one or two genes. The disability you really see, the phenotype that human patients are probably most interested in, is a learning disability. And so we did this in mice. We asked whether or not we could intervene in the epigenome, which is possible. Okay, so I'm going to back up a little bit. One of the consequences of the epigenome changing from cell type to cell type and over time is that it's like plastic. It's open to changes, right? And if we think that the epigenome causes the phenotype, it might be possible to intervene in the epigenome and correct the phenotype. So in this work here, we showed kind of what I would call short-term rescue, which means we treat them and then we measure them pretty soon after. And we do it in mice. You always have to be a little bit wondering, like, how are they measuring learning disability in mice? But there's sort of a very complicated, time-consuming assay where you try to train the mice to do something, and then you see if they become better at doing it. We have intervened in this with two different sort of broad categories. One is a set of chemicals, or a specific chemical, an H-stack inhibitor, which is a chemical that kind of goes in and hits chromatin. And another intervention we have used is a dietary intervention, which seems a little crazy. But a ketogenic diet is actually being used to treat epilepsy in the clinic. We have a whole clinic over at Hopkins that specializes in deploying this to kids. It's pretty hard to go on a medically prescribed ketogenic diet. So from a conceptual point of view, I mean, this is of course exciting, although it's in mice. From a conceptual point of view, this is important because it shows that this developmental defect can be rescued in adult mice. And that's not straightforward at all if you think about it. You get born with a genetic defect, you grow up, you develop, you grow up, and you reach adulthood, and perhaps at that point there's nothing to do about your defect, right? But here, sort of conceptually, at least in the short term, we can rescue these things out. Okay, so now I'm going to go back to a little bit sort of basic research thing and ask and show some of our work along the lines of the question: is there really a shared molecular basis for the phenotypic convergence in these Mendelian disorders of the epigenetic machinery? So the idea, right, is that they have a shared phenotype. So there should be, when you knock out these causative genes, there should be at some point in time some convergence. There should be some common disrupted features in some cell type at some cell point that causes these common phenotypes. So our idea for this work here was to look at 3 different disorders at the same time. We picked 2 disorders that was very close, KS1 and KS2, which is Kabuki syndrome 1 and 2, which are very similar, as the name implies. And then we took a slightly more distal disorder, Rubinstein-Tabi, and we did 2 experiments. So that's not like perfect, but it's something to do with like the genetics. We took some B cells, we measured them with attack and mRNA expression, and wrote it up in 2 papers. This was funded by a Hopkins Discovery Award that Hansen and I applied for a long time ago. We are not exactly the fastest people. So how do we do this? We do these experiments, so what we then do is we basically we look at the mutant mice or the affected mice, and we look at the controls, we compare them, and say we do differential accessibility or we do differential expression, we get a list of differentially expressed genes. And the simplest way to then proceed is to look at which genes are present in all the list. If you do that, the overlap is actually not that strikingly big. I mean, it's definitely there, but it's not that striking. So we thought that perhaps, we had an issue with power, perhaps we had an issue with like effect size and so on, and came up with this sort of, pretty simple method for looking at this. It's pretty ad hoc, but we've used it successfully in a lot of other contexts since then. So what we do is we take one experiment. In this case here, it's KS1, and it's KS2. And I look at the distribution of p-values in that one experiment, conditional on the p-value in the first experiment. So we usually like to think of in genomics that the p-value has this like very specific distribution. It's going to be a mixture of a uniform distribution and a peak around 0. And how big the peak is around 0 tells us how many genes should be differentially expressed. So what we see here on the left is that if I condition on having sort of, quote, reasonably low p-value in Kabuki syndrome 1, then I see a very strong peak in Kabuki syndrome 2, suggesting that there's a lot of differential expression. But on the other hand, if I condition on having a high p-value in the KS1 experiment, I basically get an approximately flat distribution, suggesting that there's really nothing happening in there. I mean, you might squint a little bit and see that the blue line has a tiny sort of negative slope, so perhaps there's a little bit, but this really suggests an astounding amount of overlap between the two experiments, much bigger than the Venn diagram is. On the left, we sort of do the same thing with Rubenstein-Tavy, which is a little bit more distal. We see, again, a strong overlap, but we also see some disorder-specific defects. This is a little bit harder to interpret than I'm making up here, because if you go back to the experiment, we had like 2 experiments. We did Kabuki syndrome 1 and Kabuki syndrome 2, a shared wild type, and we have a different experiment. So, in some sense, Kabuki syndrome 2 and Kabuki syndrome 1 and 2 should be kind of more correlated because of the experiment. So it's a little hard to kind of nail down exactly, but I would say this is sort of pretty strong suggestion. At the end of this work here, we concluded something that has been writing me as a mayor ever since, which is basically that we think that these MDEMs here are effectively should be thought of as complex disorders with respect to molecular pathogenesis. So what does that mean? I'm going to unpack that a little bit. So complex disorders means that they're going to have a lot, you knock out the gene, for example, the KS1 gene. Complex disorders means that there's going to be a lot of downstream changes when you knock out this thing out. And a lot of and because they're complex, a lot of downstream changes, and a lot of those changes are small. So that's sort of a nightmare situation, right? This is why we have a hard time sometimes making progress in a lot of common disorders with genetics, because there's a lot of hits, and the hits are really small, so we need like millions of dollars, and we're just like a couple of people at Hopkins, right? We don't have like millions of dollars, so we don't really know how to, we're definitely gonna bang our head against this. So we've followed this up a little bit right now, sort of if you should talk a little bit about present, you're sort of taking this, done conceptually a little bit similar, but on steroids, where we have used, we are analyzing data right now, we have used CRISPR to knock out 60 different EM factors instead of just 3 in neuronal progenitor cells. And then we have differentiated the cells in a dish and observed these cells at 4 different time points. So we can see kind of what happens during differentiation from progenitor cells, which is sort of a type of neuronal stem cells, into differentiated neurons and astrocytes. So the analysis is ongoing. This is really done there's a lot of people involved in it, but sort of the leads are Katrin, who is in Iceland, Katrin Müller, and Fruus, who is doing some analysis here at Hopkins. I can highlight one little figure that's a little hard to explain, but I'm going to try. Let's see how much time we do have some time. So here we have 60 EM knockouts, and each point on this graph is a knockout. I'm going to explain the diagram on the left where it says ASD. So we do a differential expression analysis where we ask what genes are differentially expressed on the knockout, and then we take the differentially expressed genes and ask, are these genes enriched for genes associated with autism? And then we quantify this enrichment with an odds ratio. We do like a physics-exact test to see if it's like significant. And what's really striking is that most of them are significant, but every single gene we knock out has an enrichment in sort of downstream consequences that also are involved in autism. That's also true for a bunch of other things. We try to come up with sort of some negative controls. Yeah, we looked a little bit at neurodegeneration, but it's kind of actually hard to find really good negative controls for this class of genes here because they just seem to be involved in a lot of different disorders. Okay, I'm gonna switch gear and talk about the last thing, which is something we are cooking. It's not really cooking, it's accepted now. It's been cooking for quite a long time. That I'm gonna call epigenetic inheritance patterns. So in this work here, we are going to think of DNA methylation as a trait, and we are sort of going to study the inheritance patterns between animals of this sort of high-dimensional trait. Conceptually, this is totally the same thing that Gregor Mendel did hundreds of years ago when he looked at phenotypes in pea plants. We just have DNA methylation, and then we have mice. This work was made possible, like a lot of work by some technological advances, particularly long-read sequencing or Oxford nanopore sequencing, where when we sequence data now, we not only get very long reads of DNA, which is critical for what we are doing here, we can also read off DNA methylation for free. So in the future, everybody who does nanopore sequencing is going to have DNA methylation for free. So that's both exciting and a little bit daunting because a lot of people are going to look at this data in blood samples, which are perhaps not so useful. So we set up a breeding experiment here. So now we're going to talk a little bit about mouse breeding. So every organism or every human and mouse have 2 copies of each chromosome. We have 1 copy that comes from your mom and 1 copy that comes from your dad. Okay, we're going to start off this experiment with inbred mice. Inbred means that the 2 copies of the chromosome are identical. Okay, so here we have 2 different strains, the blue strain and the orange strain. And the blue strain, you have 2 copies of each chromosome, and they're identical. They're both blue. And the other strain, they're both orange. So then you breed these 2 mice. You form what is known in genetics as the F1 generation. And since you inherit 1 copy from your mom and 1 copy from your dad, you're going to have 1 copy that's blue and 1 copy that is orange. So F1 mice are genetically reproducible. They're also heterozygous everywhere because the blue genome and the orange genome are different. Then you can breed them 1 more time, and then because of recombination and crossover, you get this sort of pattern where each chromosome is a mixture of blue and orange stains. So because we have long-read sequencing, one of the things we can do with long-read sequencing is we can phase the data. We can take the data, and then we can figure out what does DNA methylation look like on the blue chromosome and what does it look like on the orange chromosome, even though they come from the same cells in the same animal. We can also do it on the F2, but it's all a little bit more complicated. So here's some data, which is a really textbook example of something called a cis-MEQTL. So out to the left, we have sort of the inbred generation. That's the orange and the blue genomes, which are now purple and green. And we have a place on the genome where DNA methylation in these genomes are different. So there's a big difference between the blue and the green and the purple line inside the light pink rectangle. On the right, we have F1 mice, but where we have phased the data. That means we have taken a bunch of mice and we have sequenced them and we have sorted out for each read, does it come from the CC19 allele or CC37 allele. So each mouse contributes 2 lines on the plot to the right, a green line and a pink line. And what you can see here, and then we have the fat line. The fat line is just the average of the lines. What we can see here is that in the F1 generation, the methylation at this loci is completely determined by whether or not the line is green or pink. We have this little cartoon here down at the bottom, where at the top we have sort of the green line, and the filled out lollipops stands for sort of methylated, and we have the empty lollipops at the bottom with the purple line, which is low methylation. And we now see that we see in the F1 generation that whether or not you are highly methylated or lowly methylated is entirely determined by what copy of the chromosome you're on. So this is called a cis-acting MEQTL. And through some combinatorics and so on, you can basically show that this here means it's completely determined by factors that are local to the chromosome that we are studying. So you can also look at this in the F2 mice. So here I've taken the two, on the left I've taken the two pictures I just had, and I put them on top of each other. So there's a dotted line and a pink line. And you can see the dotted line and the pink line are basically on top of each other. We knew there would be a lot of these CSC MQ cells. They have been described for many years, but this is like amazingly beautiful data. Like this is like so much better than I think anybody expected it to see like before we did the experiment. And in the F2 generation, where we have recombination across the genome, we see that you're entirely determined by your local sort of DNA structure. Because so on the right for the efficientos, you know that we have different recombination patterns in different F2 mice. So what we really did was we bred a lot of F2 mice. We genotyped all the F2 mice, and then we sequenced 19 F2 mice, where we checked that at every location in the genome, we have at least one mouse who is homozygous AA, one mouse who is homozygous BB, and one mouse which is heterozygous. So that gives us, that's important for reasoning about transacting factors. Okay, we also discovered something new, of course. This is sort of a classic Mendelian loci. One of the things that we were really surprised to find is something called paramutation, which is a little crazy. Paramutation is something that's been well described in plants, but there's a lot of weird stuff happening in plants. It's never really been described in mammals. So paramutation in this case here, where you start off with the inbred being methylated and unmethylated, depending on what loci it is, and then they breed, and the F1 generation becomes fully methylated. That's actually not surprising. There's a lot of different mechanisms where that can happen. The thing that's really crazy is that with a paramutation, you have sort of the orange, the pink allele has gained methylation by being exposed to the green allele. And now this is inheritable. This is heritable in later generations, no matter what happens. It's become sort of epimutated. Another pattern that gives rise to the first thing in the bottom is sort of a classic dominant transacting QTL, where you again see the F1s being methylated because you have a dominant transacting factors being expressed. But in that case, once you go to the F2 mice, there are gonna be some mice, if you have enough mouse or you think a little bit about the breeding strategy and you genotype them like we did, there's some mouse that's gonna be homozygous for the transacting factor, so the loci becomes fully unmethylated. So this is a little bit bananas, and this is an example of we didn't find very many of these, but this is an example of a paramutation. We can see that the pink line, is by itself, sort of in the middle. The two green lines and the dotted pink line is down at the bottom. And when we go into the F2 generation, all the mice look like this. We, of course, dug a little bit deeper, and I kind of cut it out a little bit because it got really technical, but this is sort of caused. I'm a little bit careful with using the word caused in this talk here, but this is sort of associated by the presence of an allele-specific mobile element called an IAP in CC37. It's only present in one of the two genomes, which then becomes differentially methylated after it gets exposed to the other allele. IAPs are these weird mobile elements, and sort of real epigenetics aficionados will know that an IAP is also involved, but in a very different way in the famous agouti loci that everybody loves to talk about in epigenetics. Okay, so we're getting this out now. We have a couple of other really interesting stories, but this is probably sort of the most surprising thing to sort of basic scientists. So I'm gonna end here. I'm gonna do a little bit of thoughts and sort of acknowledgements. Okay, so first I'm gonna say is that when I work on this, I'm really happy to think that, or I'm pretty convinced that I'm kind of studying an objective reality. We are really trying, I think, in science to sort of approach the truth, right? Sometimes some people are driven by papers or recognition or money, although I do love to get paid, which you should pay attention to, which the deans should pay attention to. But we're really trying to kind of get to the truth, right? Which is sort of really kind of a goal with no compromise, which I really appreciate. And it's something that I'm often thinking about in this world here, where we almost have kind of a post-truth scenario. I feel truth is sort of losing its value. but it's really what we are aiming for. And in that process, I think it's been quite nice to work on sort of molecular biology because we have experimental tools, we have things we can go on and check, we have a relatively fast, quote, fast feedback between sort of claims and papers and follow-ups. And of course, we're not always right, but the field kind of moves forward, right? This thing that we can go out, we can create some hypothesis, and then we can actually do an experiment in real time and see what is happening. We also have some challenges. It's not always so easy to interpret what comes out of it. Sometimes experiment fails and so on and so forth. But this is really something that has been very important to me as a scientist. And it's something that I really feel you feel much stronger when you pair kind of statistics with some kind of domain level science where you actually have like some experiments that goes on here. So we often, in statistics books, we often have this idea that, the statistician sits behind his table, the collaborator walks into the door, has a very well-defined question, and the statistician sort of helps set up a model, gives him a p-value, and sends him out, and goes back to like think deep thoughts. And in my world, that's a very limited view, right? That's sort of very much like you have a client, you're helping them just with the statistics. What I've spent a lot of time on is sort of engaging in biology. I had to learn some biology. And sort of become engaged in the science to really like help with this more complicated thing that I would say George David Smith calls sort of triangulation, but you're taking, evidence from multiple different experiments or lines of research and kind of synthesizing them in order to kind of converge a little bit closer to the truth. Of course, there was an article in The Onion yesterday that sort of hit home, right? Because I've also learned some biology. I've learned enough that I know that I can be a little bit dangerous. And I don't think I'm saying anything new here to people in the Department of Biostatistics at Hopkins. We have a lot of people who are pretty engaged in domain science, but I don't think this is sort of broadly enough appreciated across our field. Another analogy I want to highlight is that we do biology. It's a little bit, one analogy I like to think about is if you're trying to understand how a car works, and we try to understand it, we try to understand how it drives, right? This is sort of the cell, and we can kind of knock out various things, right? We can knock out the window, and we can say, oh, it still drives, so the window doesn't matter. We can knock out the brakes, and then we can say, well, it still brakes, and it can also stop because, we can let go of the speeder, so clearly the brakes are not really essential for driving. If we do the experiment on a straight road, right, we can just remove the steering wheel, and it seems to work pretty well. It goes like in a straight line, and it stops. And then we can like knock out the axle, right? And that really brings the car to a halt, right? And then we can repair the axle and the car can drive again. So clearly the axle is sort of the master regulator of cars, which is of course a little bit laughable, right? I mean, the truth is we have a very complicated system. It's sort of hard to say what is sort of the most important thing. But I think this is something I often think about when I interpret sort of molecular biology experiments, and especially when I interpret causality. Okay, so I'm going to give some thanks, and that's sort of in some sense the most important part of the talk. First, I'm going to thank my family, especially my wife Margaret, and also my colleagues in biostatistics, genetic medicine, BME, and of course the rest of Hopkins. And particular to my two advisors, Andrin, who advised me, was my PhD advisor, and Rafael Lirisari, who was my postdoc advisor, used to be here at Hopkins. Then I joined, then he got the hell out of Dodge, and went up to Boston, where he is the chair at Dana-Farber and also is at Harvard Biostatistics. Although the new ratings came out, and we used to kind of share the #1 spot with Harvard, but now we're #1 and he's #2. So, if you're really into causal analysis, you can interpret this with sort of a causal angle, right? There's a clear time thing, okay, so I presented some specific work in this thing here. The little vignette on the NB compartments was really just me and Jean-Philippe. The work on the Mendelian disorders of editing machinery was done together with Hans Bjørnsen, and the main people who led this stuff was probably Leandros, Teresa, Catherine Afrous. A lot of other people were involved in this. I'm going to give Jill here an honorable mention. I don't think she was involved in any of the data I showed, but she's sort of a close collaborator in this sort of line of research. On the epigenetic inheritance patterns, most of the work was actually done by Adam Davidovich in close collaboration with Andy Feinberg, who is a long-term collaborator of mine, and David Threadgill, who is a mouse geneticist at TAMU. I'm going to give some honorable mention to Philip Wolfritz, who is one of Andy's old graduate students, Ben Langmead, who's sitting here, and also Raphael Irisari, who all helped sort of develop the computational methods that I didn't talk about, that to some sense led the foundation to all of this phasing and analysis that we did. Philip and Ben worked with me on like comparing DNA methylation across different genomes, which is actually kind of hard to think about conceptually. I'm also sort of outside of the stuff in this talk here. There's like members of my lab over the years. I've been lucky enough to work with a bunch of wonderful PhD students, postdocs, master students. I also have a couple of honorable mentions. These are people who were really working with different advisors, but who have spent many, many hours in sort of one-on-ones over a long period of time to like help them analyze the data and so on. So they almost feel like they've been contributing to my lab. There's some key collaborators that I have here at Hopkins. Andy Feinberg, who I worked with for a long, long time, Hans, Jill, and Winston, who I all work with on epigenomics. On the computational side, I've worked a lot with Jeff, Ben Langmead, who's here, Stephanie, who is also here somewhere. And in a line of research that I didn't really cover, that is more about sort of transcriptomics and particular sort of brain transcriptomics these days, I want to mention Loyal Goff, Genevieve Stain O'Brien, and then Kristen Maynard, who is at the Lieber Institute. And then, of course, all the other people I worked with sort of more transiently over the years. Okay, so thanks for listening. Oh.
Kasper, thank you for such a great talk and for reminding us about how important truth still is. That was one of the things I will take away from your talk today. I want to open it up now to questions. We are recording, and for people on Zoom, I want to encourage folks in the room, if you have questions, to use the mic. So maybe we'll invite you to come over here. And also, we'll be checking online for questions that are coming up, and we will allow you to ask those too. So please, questions, come on up to the mic.
I can't stand silence. Well, first of all, congratulations. One of the things I am inspired by in your work is the way that you've contributed a scientific understanding to the Department of Biostatistics. You're really a critical member of the faculty, and it is your willingness to become a scientist in a particular area, as well as a statistical expert, that is really inspirational to all of us in the department. And I want to say, I think the department is a special place in that people like you prosper at Hopkins, but also in the Department of Biostatistics. There aren't a lot of departments where the kind of in-depth research that you do would be appreciated the way it is here. So thank you for all the great work. I just wanted you to tell us a little bit about your experience in the department. I know there must be times when you're deeply ensconced in some epigenetic issue, and you hear people around you talking about things that must seem quite silly, but what is it like?
Well, I mean, I don't know. I mean, I think it's been wonderful to be here over the years, right? I mean, I think I've had some wonderful colleagues. We've had a wonderful social environment. I felt pretty welcomed. Perhaps, we are all sort of special in our own ways in the department, but I'm definitely, there are definitely traditional biostat departments where I wouldn't fit quite as well in as I have done here. And, I love that we are trying to think about new things to do with data, and I'm sort of excited about new things. It's super inspiring to talk to people. Yeah. I don't know, I mean, I think it's been super wonderful to be here. I think we are sort of probably the best sort of data science biostat department, at least in the US. And that's the kind of place I want to be. So, and I think that's, of course, due to the environment, right? That's caused by, one of my colleagues, all of the students, our chair, our leadership, our former chair, Karen, you, although I was never employed under Scott, and Liz. Of course, I want to thank them for not firing me yet. Yeah.
Well, Kasper, as a non-bio staff, first, I want to congratulate you. Congratulations on your amazing work. I have to admit, many of them actually I don't understand. I want to thank you actually, for many of you, you don't know, actually, Kasper was instrumental for our team. That was 10 years ago, right?
Yeah.
About 10 years ago, Kasper was very instrumental to help a team to get into the field of epigenetics from population perspective. And and also, I remember one conversation you told me, say you are not clear. I was giving a talk at the pediatric grand round. You went and told me I made a mistake. The difference between intergenerational versus transgenerational differences in epigenetic transmission. So, I was so excited you're still in this field, and we are still pursuing. So, my question for you is, are you working on transgenerational transmission?
Well, okay, so that's a complicated question. So in one sense, the last vignette I talked about was basically transgenerational inheritance, right? It was actually inheritance patterns that arises that arises in sort of future generations, right? But in that work, we are really thinking, and I think that's the best way to think about it, we're really thinking about DNA methylation as being established by the genome, like any other trait is. Transgenerational epigenetics is sort of this idea that information is transmitted through generations through sort of epigenetic factors, as opposed to being, established de novo in the new generation. And that's just really hard to show. Like, it's really hard to work with. It's really hard to operationalize. It's very hard to know whether or not when we see a factor in the downstream generations, we see some pattern. Is that because the methylation or whatever factor you're looking at was inherited, or was the DNA inherited and then the DNA established the thing? We also know that we have some negative findings, right, in mammals, where we know that, at least for DNA methylation, there's sort of these patterns of erasure of DNA methylation marks, although there are some exceptions during early development. Actually, one thing that's sort of interesting, if we talk about this, so this is a little bit an example of a transgenerational epigenetic inheritance. We know that these IAPs, or we believe that these IAPs in humans are protected against sort of erasure in the germline. So this might be transgenerational. I think I'm a little reluctant to sort of take it into I think there's a lot of people who are very excited about transgenerational epigenetic inheritance, but I think the evidence is like pretty weak in mammals. And I think it's really hard to establish it sort of rigorously. And I also think that there's a lot of people who really want it to be true because it's kind of interesting. So it's kind of a little bit the idea that you can, for example, have a methylation pattern that gets established through an environmental exposure, and this then gets transmitted to your offspring, which is quite provocative, right? And it's sort of easy to say, but it's very, very, very hard to prove. We also know that, to be fair, in some organisms, for example, C. elegans, we know that there are some epigenetic patterns, not DNA methylation, because C. elegans doesn't have methylation, but other patterns that are inherited transgenerationally. But I mean, there's a lot of weird stuff in weird organisms. So, I mean, I'm not. I mean, if you ask me about mammals, I think the evidence for transgenerational inheritance is like quite weak in general.
Well, yeah. Thank you. Congratulations again.
Thank you.
Thank you. All right. I know that we're at time. Any questions online or? Okay. All right. Well, I want to add my echo of congratulations to you, Kasper, again. Thank you for sharing your work with us, and thank you for reflecting on your work and career. And I applaud your many contributions and look forward to what else you will continue to do. Thank you all for being here today. I also want to just say how pleased I am that your family was able to be here and also online. So please join me one more time in celebrating Kasper. Thank you.